Morning,
Morning,
sir.
sir.
Morning, sir.
Can you hear me clear? My webcam, something happened to it. I've tried to add another old webcam. Can you hear me?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Okay, let's move on. So we're going to continue influenza virus from last week. What I thought is maybe we'll just have a quick recap. All of you know this. Influenza viruses are negative sense RNA viruses. They have 8 segments. I told you about the function. What is the function of the hemagglutinin? Quickly, anybody? Guys?
So, it aids in the viral attachment to the sialic acid on cell surface.
Perfect. Okay, what's the function of the neuraminidase, which is this one right here? Where is it labeled here? Okay, here it is labeled. Okay.
So it cleaves the sialic acid and reduces the viscosity of the mucus film.
That's correct. That's the one that we've looked at. There's one more function I told you. I'll tell you a little bit later in the next class, which I'll tell you today. So that is the function of this. And we spoke about a few things like this. What is the function of the M1 protein? Anybody wants to recall quickly and recap it for the class?
So it's important for the viral assembly.
That's correct. On one side, it has affinity for the outer side, it has affinity for the virus glycoprotein or the envelope proteins. On the inner side, it has affinity for the ribonucleoproteins, which is nothing but the RNA fragments or RNA segments that are attached to the polymerase. Right. So that's this protein. What does the M2 protein do?
So it's an ion channel protein which helps and aids in viral uncoating.
That's correct, because it selectively allows H plus ions inside the virion when it's inside the endosome and leads to disassembly or separating or of the fragments, denaturation of proteins, separation of fragments, so that the virus can start to replicate, which is disassembly is an essential step in virus replication. Now, what does NS1 do? I told you about that. It also reduces the interferon response, right? That's what we looked at. And I told you this is what it normally looks like, the inside of your trachea. This is in 3 days. This is in 7 days. It takes about 4 weeks to get back here. So we looked at this. We also looked at a few other things, and we looked at the role of interferon. How does interferon help? Anybody?
It induces an antiviral state in the neighboring cells and warns them of a.
Yeah, that is absolutely correct. What are the proteins that are involved? There is a 2' 5' oligoadenylate synthase which activates a ribonuclease which cleaves mRNA, and this protein kinase R is made and kept in large quantities inside the forewarned cells that are awaiting the virus, and when the virus enters, there is double-stranded RNA, and it inhibits protein synthesis in the cell by blocking or phosphorylating or making inactive an important initiation factor that is essential for protein synthesis. So cleaving of RNA is one aspect, stopping of protein synthesis is another aspect. This is what forewarned cells do to deal with viruses. Okay, so this is where we were at, and let's get started with today's class. This will continue from there. Okay, so interferons are very important protein, especially in context of influenza viruses. They are very, very important, and they are made only when they need it. They're not made and stored inside cells. They are made just when they need it. They're made when a virus infects the cell, that's when it's made. It does a lot of things. We haven't even understood much of it, but few things that we understand well are it improves antigen presentation. Now, what do we mean by antigen presentation? So there are a variety of cells which can present antigens. For example, there are some cells called macrophages or derived from macrophage lineage. So these are cells that can actually process the virus and then hold out pieces of the virus on the cell surface and call for T cell help and B cell help, so that they come and recognize the components of the virus, which are peptides, and mount an immune response. So those are called antigen presenting cells. So these antigen presenting cells, their function is improved. The ability to present antigens is also enhanced by interferons. They also do these two things. What are CTLs? Anybody wants to quickly tell me? We've discussed this before in this class.
Cytotoxic T lymphocytes.
Cytotoxic T lymphocytes. Yeah, that's correct. That's correct. Now, what are these cytotoxic T lymphocytes? Why are they called so? So these are a certain type of T cells. They have CD8 as the marker on the cell surface. Now, for those of you who don't understand, it's just a protein on cell surface that marks these cells, or that is used to identify these cells. Okay, so what do these cells do? Why are they called cytotoxic T lymphocytes? They are T cells. That's why they're called T lymphocytes. That part is obvious. Why is the cytotoxic tag? Why is it named like that? The simple reason why it's named like that is that these cytotoxic T lymphocytes have the ability to identify infected host cells and just kill the entire virus infected host cell, not just the virus, because it can't selectively kill the virus. It kills the entire virus infected host cell. That is cytotoxic T lymphocyte. Along with the virus, the host cell is also killed. So this can also lead to host cell damage. For example, if the lung cells are infected, if you have a very strong CTL response, more and more lung cells will be damaged. Now, what is natural killer cell? Anybody knows what are these cells? Okay, natural killer cells, I'll just keep it very simple. They are another type of immune cells which are, go ahead. Go ahead, somebody wanted to say something.
So it specifically kills the cell. Like they utilize perforin enzyme mechanism to kill the.
That's right. This also kills virus infected cells. So the function of natural killer cells is very, very different from cytotoxic T lymphocytes. Cytotoxic T lymphocytes specifically recognize a virus infected cell and then kill the cell, host cell along with the virus. That's what they do. But natural killer cells have a slightly different mechanisms. They can't really sense if it's virus infected, bacteria infected, what exactly is happening, but they rather work by this mechanism of just finding out unusual surface markers on the cell surface. If some markers are missing on the cell surface, they just kill the cells. So there are some markers, or there are some proteins that are present in pretty much most of our cells, and NK cells have the ability to just scan the surface. And if they see that this protein is missing, they don't really see. When I say scan, they don't see it or anything, but what they do is they try to do something called like an extension of some surface protein of NK cell and another surface protein of a normal cell. When they meet, NK cells understand that everything is normal. When this protein is missing from the normal cell to interact with the NK cell understands something is wrong with the cell. Usually, it's a class of proteins called MHC, and specific types of MHC, and NK cells have some specific surface proteins which interact with this. And when this is happening, the NK cells actually understand that something is missing from the cell surface, so something is wrong. It may be virus infected, bacteria infected, parasite infected. It may be a cell that is becoming cancerous. So NK cell just kills the cell. So that's why it's called natural killer cells, because it just kills anything that it thinks is unusual. So that's NK cell. That is also important here. There are many, many more downstream things that happen because of interferons. Most scientists think that we have understood a very small tip of the iceberg of what interferons do for the body. But its interferons are critical to eliminate influenza virus infections. Now, what kind of disease does it cause? Usually, in most people, influenza is uncomplicated. So there's fever and chills and headache, cough. All of you know this. Myalgia is muscle pain. So who are the people who have pulmonary or lung related complications with influenza viruses? Usually it's the ones who are like 65 plus, or the ones who are less than 1 year old. And pregnant ladies. So these above 65 comorbidities exist. Immune system is slightly weak in pregnancy. Actually, there are a variety of hormones in pregnancy that are produced in large quantities that alter the immune system and tilt the balance between Th1 and Th2 response. So pregnancy is another kind of immune response. We will look at one virus disease where pregnancy changes the mortality rate like by 25-fold. It increases the mortality rate much more than 25-fold also. So we'll talk about that. Pregnancy can affect the way or the kind of immune response one can mount. Anybody wants to guess why less than 1 year olds could have more complications? Nobody wants
They
to
haven't
guess?
been exposed to that many diseases, so their immune system sometimes gets hyperactive.
Let's say you're also not exposed. I'm also not exposed to 1 year old also is not exposed to that particular virus. We will all kind of respond in the same way. But what is peculiar to 1 year old would, apart from the immune system, I think immune system generally more or less would by in a few days of birth, it becomes robust. Okay, so I'll give you the answer. So everything has to do with the airways, right? And the bronchi, bronchioles, and the airways. So let's say this is the airway, the path through which the air goes inside our lungs. So usually the problem with why a lot of respiratory infections, for example, RSV, respiratory syncytial virus, and few other viruses, respiratory viruses, cause more severe disease in less than 1 year olds. It's because of the size of the airways. So under 2 years, the airways are very narrow. So let's say this is the size of the normal airway, the 1 year old airway. Let's say this is the size of 3 year old airway, and beyond that, it kind of stays, let's say, more or less the diameter remains same. So let's say if there is inflammation here, there is also inflammation. This is both sides of the inside of the airway. So if there is inflammation here, the same amount of inflammation here will still let air pass through, but here it creates more problem because the airways are narrow and that size becomes reasonable. Usually after 1 to 2 years of age, it becomes broader, and that allows that accommodates some amount of inflammation. That's why it's difficult for less than 1 year old. So then they can have virus related pneumonias or bacterial pneumonias or secondary bacterial infections. Anybody wants to say one line about influenza virus and secondary bacterial infection that we looked at in the last class? Nobody?
So when there will be no cilia, then there is a chances of these bacteria to infect the respiratory tract.
That's correct. So one of the important things that happens is this mucus layer becomes less viscous and the cilia is removed where yeah, like this. This top layer is lost. So they don't have the ability to trap and expel pathogens or some pathogens that may be there in the throat in small quantities. So that is one of the reasons that secondary bacterial infections. Why are they called secondary bacterial? Because they're secondary to the virus infection, which is a primary cause of the disease. The virus infection is the one that actually clears off the top layer of the inside of the respiratory epithelial tract and reduces mucociliary clearance, leading to this bacterial infections. So that's why virus infection is primary, and in this case, bacterial infection is secondary. So many times actually after influenza virus, secondary bacterial infections are very common. So why is influenza virus why is it a little bit of a threat? Of course, because there is a mortality rate that's associated with the virus. That's why it's a bit of a threat, but also because it can cause non-pulmonary complications. What do we mean by that? It can cause complications outside the lung and the respiratory tract. For example, one of the things it can cause is muscle inflammation, but that is not life-threatening, right? So, but this part and this part are serious. What do we mean by these two? So it can cause cardiac complications in a lot of people above 65. any pre-existing heart conditions, it can worsen. And recent studies have shown that actually it can infect cardiac cells or the cardiac muscle cells. It has the ability to infect. Not only that, but very recent studies have also shown that it also interferes with the transmission of impulses that control the heartbeat. It interferes with that. So these are the reasons why it can cause cardiac complications. So influenza viruses have the ability to also replicate in some neurons, and the immune response that helps clear the infection is usually like what you saw here for the lungs. Where is this one? If something similar, so if this is the inside of your trachea, and after infection and the immune response, it becomes like this. If the same thing happens in neurons, you can imagine that there is going to be a lot of neuronal damage and related neurological complications. So these are extrapulmonary complications that influenza virus can cause. So there is a small percentage of people who can succumb to influenza viruses or die because of this. The most common cause is not usually the viral infection or the viral pneumonia. It's more often than not is the secondary bacterial infections that cause pneumonia and kill the person. Above 65-year-olds, they can have cardiac failure, and less than 1-year-olds, because their airways are small, they also have higher mortality rate. The same amount of inflammation will affect them more, in other words. Okay, what about the immunity in influenza viruses? Which is the most important immune response? I've told you that several times. Anybody? Anybody wants to tell me what is the most important immune response in influenza virus? Guys? You guys with me?
Yes, sir.
Which one? Let's be more interactive. I was just screaming my throat out about interferons. Okay, so interferons are the most important immune response, although there are other types of immune response, which is antibody mediated immune response, which is important. Okay, two things from this slide. Which is going to be the neutralizing antibody? All of you understand what is neutralizing antibody. Neutralizing antibody will prevent the virus entry into a susceptible host cell. That's a neutralizing antibody. What does that mean? If this is a respiratory epithelial cell, which is what is written here, it has the right receptor. This is the virus. But if you have antibodies that can bind to the hemagglutinin, that is shown here, they will block the interaction between hemagglutinin and the sialic acid receptor, thereby preventing virus attachment, virus entry, and this antibody is called neutralizing antibody. So if you have antibodies to the correct, to the hemagglutinin, it will actually prevent infection. That's what this antibody will do, and that's why this antibody is called neutralizing antibody. Now, on the right side of the cartoon, there are two aspects of the antibody to the neuraminidase. Left side is influenza virus hemagglutinin, right side is the neuraminidase, right? The antibodies to the neuraminidase. There are two aspects of this I want you to understand. The first aspect is that antibody to neuraminidase does not prevent infection because it cannot block the step. Can it block the step? It cannot block the step. Is everybody with me? Can you understand that neuraminidase antibodies will not stop infection?
Yes, sir.
Okay, so on the right side of the cartoon, we'll go on to the second part that I want you to try and understand. Let's say we are all vaccinated against influenza virus and we get infected. Okay, the vaccination usually has a good protection. It will reduce the severity of disease because very few viruses can attach and enter. This step is blocked, and also because our vaccine, the vaccine for the influenza virus usually contains two important components, the H and the N, as you all know it, hemagglutinin and the neuraminidase. These are the important antigenic epitopes that will provide us immunity against influenza virus. The antibodies against H or the hemagglutinin will prevent infection, whereas the antibodies against neuraminidase will not prevent infection. However, they are helpful in reducing the severity of disease. How does this happen? So let's say this is an infected epithelial cell, respiratory epithelial cell that's infected with influenza virus. Like I told you, we looked at how the virus replicates and how the hemagglutinin and the neuraminidase come and sit on the cell surface and are waiting for the virus with the capsids and the matrix proteins to bud out of the cell. And the matrix protein has high affinity for the hemagglutinin and neuraminidase, so it sticks to it and the virus comes out. So when the virus comes out, because this is the cell from which the virus is trying to bud out, the virus has neuraminidase right here. The cell surface has neuraminidase right here. Let's say we have antibodies against neuraminidase. It will bind to this part here, and it will also bind to this part here, and this will actually prevent virion release. So that's what antibodies to neuraminidase do. They aggregate the virions on the surface of the infected cells, and they don't let them go and infect new cells. So the burden on your lung and the burden on your immune system goes down, so that you have the number of viruses that are actively replicating goes down, because this will be actually, it will just hold it right here and cause aggregation. After that, it will just actually degrade after that. So that way, neuraminidase antibodies are important to reduce the severity of disease. Is everybody with me on this?
Yes, sir.
Yes, sir.
So overview of the immunity to influenza viruses. Usually the immunity is long-lived, which means once you're infected with H1N1, maybe you have good immunity against H1N1 for several years. That is what we mean by long-lived. The immunity is also subtype specific. What do we mean by the subtype here? Is the H and the N configuration. H1N1 is one subtype. H1N2 is another subtype. You can also write H2N1 is another subtype. H3N1 is another subtype. So it is usually subtype specific, which means the protection offered is usually against this particular subtype. If you had exposure to H1N1, you have good protection against H1N1 for a long period of time. But let's say you get H3N5, you may not have any protection against H3N5 or very little protection against H3N5. That's what is meant by subtype specific. So because it infects the respiratory epithelial tract, we produce IgA antibodies, which last few weeks to a few months, but they offer mucosal immunity or local immunity, like I explained to you in poliovirus, why the oral poliovirus vaccine stops the virus before it enters the blood, whereas the injected poliovirus stops the virus only after it enters the blood. So that is all related to mucosal immunity, and the most important antibody for mucosal immunity is IgA antibodies, like I told you before. And because we get infected, our mucus cells or the cells of the epithelial tract are infected, there will be a strong IgA response, and this is very important because it controls the virus much earlier than IgG, which lasts much longer, but it's there primarily in the blood. So this will last only for a few weeks to a few months, but this will last for several years, but this will control the virus only when it enters the blood mostly. So, right, but it has other aspects of immune response where it can help cytotoxic T cells. It can also help other types of killing in the body, but regardless of it, but primarily the role comes, it kicks in a little bit later when the virus enters the blood, whereas IgA kicks in much earlier when it's in the respiratory tract, because IgA is secreted from these mucus cells of the, pretty much most of the mucus cells in the body can if they get exposed to a pathogen, they will have an IgA response that lasts for a few weeks to few months. Is there cross protection between these three? Of course not, because there's very little cross protection between the subtypes of influenza A. There is no cross protection between A, B, and C, which are different viruses altogether. And what does this mean? Cell-mediated immunity is the kind of immunity that is associated with killing the infected cells. That predominates here, and that's why there is significant lung damage, heart muscle damage, neuronal damage. All these things happen because this is a predominant type of immune response that helps steer the virus. Now, how do you diagnose this virus in the laboratory? What do you think would be the samples one needs to collect if they want to diagnose influenza virus, just from your experience of SARS-CoV-2?
Nasal and throat.
Nasal and throat are the predominant samples. That's exactly what I have here. And typically when I was doing my master's, and like much after that, and even now in many places, there is something called tissue culture, which was the predominant way of diagnosing influenza virus. It can grow very well in embryonated eggs and in some cell lines. So growing it and then doing other tests was the norm for a very long time until a few years ago. It has all become RT-PCR. This is now the predominant method of diagnosis, or the mainstay diagnosis from this sample, of course, nasal or throat swab samples. Like all of you understand, I think everybody, like pretty much the whole world understands that why we have to take a respiratory sample and pretty much how RT-PCR works. Thanks to SARS-CoV-2, I think everybody in the world understands what this is. Now, before the advent of RT-PCR, actually even 3 years ago, this was not a common test in India, but now it's becoming more common because our capacity to do RT-PCR has expanded big time in the last 2 years because of the pandemic. Now, how did people do this before 2 years ago or 3 years ago in resource limited settings? Like the very developed countries switched to RT-PCRs completely almost about 10 years ago. They switched to RT-PCRs, but we were doing it like this, growing it. Then we do something called antigen detection, or we do something called ELISAs. I'll tell you what that is in a minute. I think I have cartoons. Or we can detect antibody response to influenza virus, but that is not useful because a good number of people have been vaccinated. A large number of people may have been exposed to influenza virus without knowing. So antibodies, IgG antibodies, because they last very long. If I got infected at 10 years old, even now I may have IgG antibodies to influenza virus. It's possible. So that way, antibodies against influenza virus do not help you diagnose current or ongoing infection. They are just an indicator that you've been exposed either by means of vaccination or by natural infection. You've been exposed to the virus. That's all serology tells you. I'll tell you a little bit more about these two, which is immunofluorescence and ELISAs. I'll tell you a little bit more. But a lot of times in our country, for example, maybe it will change in the next 2 years or so because our capacity to do RT-PCR is now much bigger. But like, let's say in 2018, even today, for the most part, if you go to a doctor with symptoms like fever, body ache, and respiratory symptoms, most likely they'll do a SARS-CoV-2 test. If you're negative, they're going to send you back because they're not going to diagnose influenza viruses. That is what is happening for a long time in resource limited settings, because very few labs had the capacity to do specific RT-PCR, and also because it was not causing outbreaks or it was not causing a pandemic like the one that's ongoing now. We didn't have the necessity to test everybody. If it's uncomplicated cases, the doctor will just look at the clinical picture, and if an outbreak is going on in that region, he will provisionally diagnose you with query influenza virus and then send you back home to recover in the next 4 or 5 days. Most people recover very well. So this is what happens more often than not, but But in the developed countries, they put more emphasis on diagnosis, specific diagnosis. Even if you don't require specific treatment, they try to diagnose specifically. They try to understand the epidemiology of the disease, the burden of the disease, the need to vaccinate. All these things come from doing more and more diagnosis. If you don't test anybody, you will not have any information on what strain is circulating, how much is the burden, is it becoming predominant, nothing you'll know if you make only a clinical diagnosis like the one indicated in the last line. So for that reason, doing diagnosis is a very good thing. So, and that is done routinely in more developed countries using RT-PCR than any other method. I'll give you a quick look into how to do immunofluorescence. Let's say you get a sample, get a throat swab or a respiratory swab, like one of you rightly said. But what is done is it's put on a glass slide. You just make impressions of the swab on a glass slide. Just add a chemical and fix it. Add another chemical and permeabilize the cells, which means allow antibodies to get in. And then this antibody that you add after treating it with, after fixing it and permeabilizing the cells, is what antibody do you think you should use? Guys, it's written here, use an antibody against hemagglutinin, and these are your respiratory epithelial tract cells, either from the throat or the nose or somewhere. And if they have hemagglutinin, let's say this sample has hemagglutinin, these cells have hemagglutinin, and this is an antibody that can go and bind specifically to hemagglutinin, you add that antibody. And at the back end of the antibody, you have a label that's stuck to the back end of the antibody, the base of the Y, if you would call it. Now, this is the fluorescent label. It's called FITC, or there are many other possible fluorescent labels. So this will fluoresce in green color. So if you see green color fluorescence, that means these respiratory epithelial tract cells from the nose or the throat, they have hemagglutinin from influenza virus, which means these cells are all infected with influenza virus, which means the person is infected with influenza virus. This is the basis of immunofluorescence. This was very widely used in our country because molecular tests were way too expensive. It used to cost almost 4,000 to 5,000 rupees for a PCR. So, but it used to cost only about 1,000 rupees for this test. So this was done more often than that. Now you can also take the respiratory or the throat sample and put it into small wells called plates called ELISA plates, a well of something of this size. And inside that well, if you zoom into it, it's coated with an antibody that can bind to hemagglutinin, which is present on the virus surface. It binds to it. You add another antibody that can also bind to hemagglutinin because the virus has many hemagglutinin on its surface, and the back end of the antibody is either, it's usually labeled with an enzyme, and then you add a colorless substrate which becomes a colored product. So for example, like wherever, or an orange substrate becomes a blue product. So wherever you see the blue, those are the positives. These are the positive controls. All the orange is negative. What changes the color from orange to blue? I'm just going to clear this and tell you in a second. Many of you know ELISAs. It's the enzyme that's attached to this back end of this secondary antibody. So if you did not have hemagglutinin, if you did not have influenza virus, this will not bind. If this does not bind, this will also not bind. It will be washed off. So there will be no enzyme to change the orange substrate to the blue product. So there would be, it'll remain orange if there was no influenza virus. So if there was influenza virus, this will bind here. Secondary antibody will bind. Enzyme will be stuck to the back end of the secondary antibody. You add a orange substrate, it becomes blue. Orange, blue, colorless to colored, all these, there are like a variety of enzymes and substrate combinations. So don't worry about it, but there is a color change. And this is, as you can see, 8 by 12, 96 well plate. Each well is one sample. Each well is coated with only this. Rest is added later. It's coated with an antibody to hemagglutinin. And this is another way by which you can detect. why would people prefer RT-PCRs to this test? Anybody wants to give me a quick answer? You can do 96 samples here also. You can do 96 samples in RT-PCR also. Guys, I want you to think and tell me simple things. Your voice is breaking. Is it my internet or is everybody hearing a broken voice?
It's broken for us as well, sir.
Okay. I can't see who's speaking. It shows up as ML, but if you can speak a little bit louder, or it'll be better. We can hear you better, I think. Go ahead, please. Can't hear you. Anybody else wants to try? Still too broken. I can't make out much of what you're talking. It's your internet, I suppose. Anybody else wants to try that? Very simple answer.
So maybe because RT-PCR is like almost a short confirmation, because you actually get the sequence information.
It's not that actually. It's not related to whether it could be a false positive or not. It's more because RT-PCR amplifies stuff. If you think about the principle of RT-PCR, if you have 10 copies of the virus, if you have like even 5 copies of the virus, some RT-PCRs can amplify it like a billion-fold and allow detection. Whereas if you have only 5 copies of this, you have very little secondary antibody binding, very little enzyme, very little color change. You will miss it. The bottom line is RT-PCRs allow amplification of whatever target you have. Here there is no amplification of the target. That is the difference. Because you amplify the target, whatever DNA or RNA you want to detect is amplified, RT-PCR allows a much better sensitivity, or allows detection of very few viruses also if they are there. So they allow for better diagnosis. That is the principle of, that's why RT-PCRs are more preferred. Although this will be much cheaper than RT-PCR and requires much less training and instrumentation, the sensitivity matters. The ability to pick up low copy number samples is very high in RT-PCR. Okay, then I'm going to go on to a concept called antigenic drift and antigenic shift. Okay, first we look at antigenic drift. Just before we go on to this topic, I feel all of you are very quiet. So maybe we'll just ask a question. The English word drift and shift, which one appears to be more pronounced? Which is a small change, which is a bigger change?
Our shift will be bigger.
Shift is a bigger change.
Lower.
Just like all of you understand, shift is bigger. That is correct in terms of viruses also. So I'm going to try and define what is a drift and what is a shift in the next few minutes. Okay, so of course, all viruses mutate. RNA viruses mutate more rapidly than DNA viruses. So there's going to be gradual accumulation of mutations in the H and the N of the influenza virus as it replicates and spreads in humans or animals. It's going to gradually accumulate mutations, and that is called an antigenic drift. Now, in an antigenic drift, which is a small change, because one mutation occurs and many mutations don't stay because they are lethal. They will not be able to replicate. They are lost during evolution. Only some become fixed, and because this is accumulated gradually over a long period of time, usually the configuration of the H and the N, like H1N1, will remain H1N1 in spite of antigenic drift. For several years, it will not affect the configuration because the change is so small, right? It does not change the configuration, but there can be mutations in H and N. So what will happen is it can change to an extent where people who had the mutations that are accumulating because of drift in hemagglutinin and neuraminidase may may allow already exposed individuals to become infected again, or in other words, it may not fully protect against the new mutations that accumulate over time without changing the H and the N configurations. These mutations may not be neutralized by the antibody against the wild type strain. So that is called antigenic drift. Anybody wants to give me a good example and compare what could be drift in the current pandemic?
Sir, small mutations accumulating, so we are vaccinated against Delta Plus and again we are infected with Omicron.
That's a very good
That's
example.
maybe
That's a very good example. Whoever said that, see, basically, we are vaccinated against wild type SARS-CoV-2, and that is now not protecting us from Omicron. Omicron is not a shift. It's not a sudden change in spike configuration of coronavirus. It's not. It's a gradual accumulation of mutations that is rendering antibodies against the wild type ineffective. Did any of you get infected with Delta and Omicron also? Nobody? Okay. So there's a large number of people, even in the Kusuma school, there are a lot of people who got reinfected. So there's a large number of people who've been infected with Delta and then Omicron. That is exactly what this means. Immune response does not protect fully any longer. Although they might have had milder disease, they got infected. That's because the immune response, because the surface proteins, the spike protein in this case, has changed enough that whatever antibody you have against spike does not neutralize it completely. Same thing for H and N. The H and the N configurations remain same, but there are changes. I'll tell you what, when the H and the N configuration will change, then you'll appreciate this better. But these are all mutations. Okay, so but because there is partial protection in the community, because we had a large amount of Delta, Omicron is not spreading. Although it can spread, it is not causing serious disease, and the extent of the epidemic is limited by a great extent. So this occurs in all three influenza types. Influenza A, B, and C. I'm just using the current pandemic as an analogy so that you understand better, because none of you may understand or relate to influenza virus that easily. But it's easy to relate to Delta, Omicron, and the vaccine much more easily. So this is about antigenic drift. You'll probably understand it better when I show you what is shift. Before I go on to shift, I'll just show you a cartoon about drift, which is the smaller change again. So this is the shape of the hemagglutinin here. Okay, it's changed slightly now, although it is H1 only still, but it's slightly different that the antibody you produced against this shape, this club shape, is not going to work anymore. It's not going to work very well. So this is the concept of antigenic drift, slow mutations accumulating and immune response becoming partially protective or not fully protective. This is the concept. Now, what is antigenic shift, the bigger change? Before we understand the bigger change, I'll just introduce one more small thing about influenza viruses, which is very important. Because these are segmented viruses, they have 8 segments each. When 2 different configurations of influenza virus infect the same host cell, I'm just going to call this H1N1. Okay, just to keep it simple, I'll just call this H1N1, and I'm just going to call this H2N2. Okay, just an example. If both of them infect the cell, and let's say this is let's say this is this is H1 from the red color. This is N1 from the red color. Okay, H1 and N1 from the red color. the rest of it is all from this virus. So because when they infect the same host cell, the 8 fragments from this virus and the 8 fragments from the blue virus, they all replicate actively inside this host cell. And when it gets packaged, it doesn't have a way of choosing between red and blue. It just chooses the minimal 8 fragments that are necessary and they get packaged. So when this happens, there's going to be mix and match between the red and the blue, or this virus configuration and the other virus configuration. They're going to be mixed. The fragments are going to be mixed, or the 8 segments of the genomic material are going to be shuffled like a pack of cards, and you get a variety of reassortant viruses. And what is this virus now? It's going to be called this virus now. Although the 6 parts or 6 fragments of this virus are blue and they represent this virus, the H and the N, the 2 red pieces, they come from this virus. So now this surface display will become also different, because on the surface it will have the red colored H1N1 only will come on the surface. So this virus will now be called H1N1 virus, but it's a reassortant virus. So this is how it can lead to a new configuration. Maybe if this is H3, for example, and N2, and this is from H1. The blue is from the N. The blue is from N2. So this will be H1 N2. Okay, so it can just lead to any combination, but we don't care that much about the 6 fragments, although they are very important, and they can also determine host susceptibility to a small extent. They may determine host susceptibility. They don't determine the attachment or negotiating the mucus part. So especially the H configuration becomes very, very important. So that is why whatever is the H encoding piece, if it gets shuffled, it leads to the production of a new virus. Now, what does this reassortment lead to? This reassortment leads to drastic or abrupt and sudden changes in the configuration. So for example, okay. Can you hear me? My power is down. I don't know whether my internet is working. Can you hear me now?
Yeah.
We can hear you, sir.
Yes, sir.
Okay. Somehow I still have internet. Okay. So what it took for this configuration to become this configuration, the subtle change, it might take like 50 years of time. It's still, this is also H1, this is also H1, but they are subtly different from each other. But here it jumps from H1 to H2 or H2 to H3, whatever. It can jump to any configuration, and this can happen just in a matter of a day or two. The cell goes in, these come out. It's just a matter of like 24 hours to 48 hours, you get a bunch of viruses with all new configurations. So that's why this change happens so abruptly. And it's called reassortment because it's shuffling of fragments of genetic material. It's not a single nucleotide or a small deletion or a small insertion. It's just changing different fragments and mixing and matching. So that becomes a new configuration altogether. Just overnight, you get new H and N configurations. So pre-existing antibodies will absolutely not protect usually, because H1 antibodies will not protect against H2 because it's significantly different from H1. Same for H2, same for H3. So if you have antibodies against a particular H type and an N type, you have protection only against those H and N types. So they don't protect. And these H and N types can change overnight by reassortment of 2 viruses inside a same infected cell. And it can cause new viruses or new configurations. And this is called antigenic shift, because it is a much larger change, it's much quicker, it's a much bigger change, and it happens only in influenza A. I'll tell you why this is in a bit. And this can cause large-scale epidemics or pandemics. Let's assume that everybody had H1N1, the whole world, suddenly there is a new virus with H2N2 configuration, and nobody in the world has been exposed to H2N2. There is no protection of the H1N1 antibodies. They don't offer any protection or very little protection against the new configurations of the H and the N. They don't offer much protection at all. So the whole world is now susceptible like they've never seen influenza virus. If there is a new configuration of H and N, this is what happens, and that's why they can lead to a large scale outbreak. So this is again a small cartoon to illustrate what is shift and drift. So drift is the smaller change, like all of you understand. If it's purple colored, it remains H1 or N1. The configuration does not change. However, the shape of the H within H1, it can lead to subtly different configurations of H1 itself, but it will not change the whole configuration of the H or the N. So that is antigenic drift. Or if it has to change the configuration of the H or the N, it may take centuries or like several decades to change the configuration. Usually it does not change. Now, this is one H and N configuration in different colors, second one, third one, all of them represent different H and N configurations. When the same cell gets infected, you can have variety of configurations, new subtypes emerging. A new subtype, when it emerges, the whole world is susceptible, and this is possible because of reassortment, and this is called antigenic shift. Is everybody with me on this?
Yes, sir.
Yes.
Yes, sir.
Okay, we have only another 3 minutes or so. So I think we'll just stop here, and I'll take any questions you have in today's class. Anything that you did not understand or anything that you had questions about, you can't relate to, please tell me. We have another 3 minutes.
Sir, if there is antigenic drift, will the ELISA test will be can be done for it?
Okay, very good question. I think that's a very good question. Let me go back. So ELISA tests are not widely used, but if you were to use an ELISA test, let me try and go back. Okay, here we go. Just going to clear this and answer your question. Okay, so let's assume this is the antibody to hemagglutinin, which it is. So whenever you want to do an ELISA test, a diagnostic ELISA test, there are multiple ways. If the virus is changing very fast, you want to use what is referred to as polyclonal antibodies. What does that mean? That means you have hemagglutinin. I told you, like, one protein has many epitopes. Like, let's say if it's a protein of 1,000 amino acids, maybe it has 10, 15 epitopes, something of that order. And each epitope can elicit a different antibody response. So there are 10 or 15 different antibodies for the small protein. That happens normally inside our body. There are two options of using antibodies for diagnostic assays. One is called a monoclonal, the other one is called a polyclonal. Monoclonal means it's very specific for one epitope. The other one is specific for multiple epitopes. Polyclonal means all the 10 different epitopes, 15 different epitopes of the protein, all the antibodies will be pooled. So if you use a polyclonal antibody, that enhances the sensitivity of the ELISA. Does that make sense?
Yes, sir.
So if you take a H1 hemagglutinin 1, you raise polyclonal antibodies, which means all the epitopes, maybe about 15 epitopes of hemagglutinin. You use that here and here, then you will not miss much of any configuration of HAN1, any variants, small variants, drifts, you will not miss. But if you use monoclonal antibody that is against only one of the multiple epitopes, you may miss some. Does that make sense?
Yes, sir. But if there is any conserved domain, any conserved epitope, so in that case we can use monoclonal antibody.
So diagnostics is actually a game where you have to balance between sensitivity and specificity. So they always use combinations. Some assays use one monoclonal and one polyclonal. They use a variety of combinations. You are absolutely correct. If there is a highly conserved epitope, we can use that, and that you may not. Then you can use a monoclonal. That will stay same in all the HA1s or hemagglutinin 1. If there's a small conserved domain which is immunogenic, that is good enough. So you can use a monoclonal also. Any
And
other
sir,
questions? Yeah, go ahead.
And antigenic drift and antigenic shift can lead to serotype, new serotype.
Yeah, so okay, let me go back to this. Do I have a cartoon for that? I thought I had one more cartoon, which, give me a second. Okay, although I didn't go through this cartoon, I just showed you. This is antigenic drift, small changes. Okay, small changes. Then it remains same for some time. That spreads. After that, some small changes. This is the amount of changes. Okay, this happens. Let's say this happens in, let's say, 100 years. Just an example. Here you may have something that looks like a new HNN configuration. It is possible, but it may take several decades or like maybe even several centuries for that to happen. But here you have it like you just have it today. It changes and it infects 100 human beings. New HNN configuration. That can lead to an antigenic shift or a new configuration or a new serotype, or here we call it subtype. It can lead to that within a matter of few days. It can lead to that. So the answer to your question, short answer, whether drift can change the subtype, nobody says no, but usually it does not. But if it had to change, it will take decades or centuries. But here in shift, it can just change it in a few days. Actually, just one event, right? What I showed you, reassortment can lead to that. And after that, it's just a matter of that virus spreading to the people. So this happens very quickly. Here it happens very, very slowly. But in one's lifetime, it may not change the H and the N configuration to a new one. But we are not saying it will never change, because slow changes will amount to a big change, right? It's like 10 rupees every day will amount to 1 lakh rupees in a lottery. Eventually it will amount to that. It's something like that. Both can lead to it, but usually this does not lead to it within a few decades or it may take decades or centuries to change the configuration. Antigenic drift, the small changes. Any other questions? Okay, guys, I think we are over short time. I'll close here. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, guys.
Thank you, sir.
Thank you, sir.